All right, immunology fans. Yeah, let's go. Let's go. Part 3, Chapter 4, Part 3. All right, here we are. Um, let's see. So now we're going to get into T cell receptors, which, as it says throughout the book, they're very similar to the way they're basically similar to an FAB. They're going to consist of two chains. They're going to have a sort of a constant region at the bottom and a variable region at the top. The variable region is going to have a couple of loops on it. Those loops are going to be hypervariable, and you're going to get different versions that are able to bind to different things. Yeah, it's not any smarter than that, I'm afraid. Which I think that's a nice theme that runs through immunology. It's a bunch of mechanisms that are basically straight up not very smart, and they're kind of low percentage plays, but we run enough of these. We have so many cells trying these same low percentage plays over and over and over again that we end up coming out, in reasonably good shape. It's inefficient as heck. Yeah. Because just imagine, if you could go in, there are probably ways that you could actually say, well, all right, if I'm going to run into SARS-CoV-2 next week, then I know I'm going to need, this antibody, this other antibody. This other one's not going to bind, so we don't need to make any of that. And just order it up, order up this particular domain, these together, and if there's a way to tell your cells to do that, then theoretically we could build immunity without vaccination. But that does not yet work that way. And because the immune system works, the adaptive immune system rather, like B cells and T cells, because that works by actually changing DNA, chopping out lots of big parts of your precious DNA genome and gluing other parts together. Because it would basically take deliberate gene editing with probably higher efficiency rates than we can currently make happen in order to do that without just straight up killing people. So, yeah, it's like a reasonable research goal, but it's not something that is ready for prime time yet. But maybe in our lifetimes, I don't know. Maybe I'm being too optimistic. You never know what progress is going to bring. Yeah, progress is funny like that. So, there we go. Okay, so here's our little comparison. An antibody is very similar to a T cell receptor, except that they're different chains. So we call the T cell receptor, we're going to say that this one's an alpha chain, and we're going to say that this one's a beta chain. Like, okay, great. Whereas we called this one a light chain, and we called this other one a heavy chain. And so the alpha is going to be like the light chain, and the beta is going to be like the heavy chain equivalent. Actually, fairly precisely. So they're different genes, but they're set up the same way. They take on roughly the same shape. They're made of immunoglobulin-like folds, just like each other, and they both are there to shift and adapt and bind to antigens that are presented to them. And we'll get into antigen presentation, like what that means. But there are specialized cells whose job is to find something that doesn't look right, and they don't have to understand it. They just have to chew it up a bit and then just hold it out and say, yeah, anybody recognize this? Anybody? That's their entire life. It doesn't sound amazing. I don't think that's a good career goal for any of us to be an antigen-presenting cell, but they make the thing work. Yeah, so there you go. Just a bunch of cells doing their jobs. That's all your immune system is. Okay, on the T cell receptor, and actually on some of the antibodies as well, there will be carbohydrate molecules. So this is a chain of what are chemically sugar molecules, but there'll be a big branchy chain. And you tend to get these carbohydrate molecules on proteins that are expressed on the surface of a cell. It acts a little bit like camouflage. It stops antibodies from coming in and just sticking all over the parts where you don't want them to stick. And so basically we're just masking the sides and bottom of the T cell receptor. And we're leaving free the top of the T cell receptor, which is the business end. That's what's actually going to bind to a little bit of antigen. So, Big difference between a T cell receptor and an antibody, FAB, is that the T cell receptor is going to be anchored into the membrane. So they're still going to be held together by a disulfide bond, just like in an FAB part of an antibody. That's going to be in the little stalk region, which is just underneath the bottom immunoglobulin-like domain of the alpha, yellow, and beta, green chains. And then there's our little transmembrane region, and there's a little tiny tail at the bottom. And so the tail has some binding sites on it where other proteins can bind. For example, you'll have kinases that can latch onto this and can start to activate other proteins once the T cell receptor has made contact with the right protein. And the way it's going to know that it's done that is that when the T cell receptor is holding on to something, there's often going to be another protein that's going to come in from the side. So one of those is going to be called CD4, and you find it on helper T cells. Another one's going to be called CD8, which you find on killer T cells. And there are also it's like a rainbow of T cells out there. There are some that have both CD4 and CD8. And there are a lot that have one or the other, but have additional markers that give them additional powers. It's like a Marvel cinematic universe full of slightly different superheroes out there protecting you, and who the heck cares? Yeah, you care, I guess. Yeah, that's why you're here. Good for you. Well, we'll get into them, but it's a lot. Yeah. Okay. So this is a T cell receptor. And so they've cut off the part at the bottom where it would go through the membrane, because when you're doing a crystal structure, you need a soluble protein. So you basically have to snip off any transmembrane parts and just pretend that there would have been one down there. Totally, yeah. Because you know where it is. You just can't really see it together there. And so at the top, this 1, 2, 3, those are our 3 little hypervariable loops. 3, just like in the antibody, right? 3 little loops on the heavy chain, 3 little loops on the light chain. And so here's your alpha, which is like your light chain. Here's your beta, which is like your heavy chain. And there you go. And you can see that on the top of it, here, for example, is a little molecule that's bound there. And these are the hypervariable regions that can actually bind to different substrates. So here we show it bound to a carbohydrate, and good. They can bind to proteins, they can bind to potentially lipids, carbohydrates. And you would say, well, wait a minute, why would I want a T cell that would bind to lipids? But bacteria will often make these weird lipids. So sometimes it'll be a lipid with a sugar on it. Sometimes it'll be a lipid that looks strange. Like you're used to the kind that have 2 tails. Sometimes they only have 1 tail. Sometimes the tail is like a circle. Yeah, that's mostly an archaea. But there's strange things there and different modifications that you find in bacteria that are useful to recognize and that you will never run into in the course of a normal day's work inside of the human body, because we do not make them and do not need them. Yeah. So, and like I've said in class, probably, who knows? Yeah. The immune system is kind of a by any means necessary sort of operation. If it leads to a T cell receptor or an antibody binding on to something from a foreign invader, that's a win. Yeah, no matter how it happens, just take the win. Yeah, take the dub. All right. So here's a T cell receptor, and here it's recognizing a little antigen, and T cells specialize in recognizing antigens that are bound to these MHC molecules. So those are our major histocompatibility complex. And so there are 2 main types of these. The class 1 MHC molecules are going to be displayed on pretty much every cell in the body. And these are going to show a little piece of whatever protein was inside the cells. They'll chop up their proteins and load them up on these things and display them on the outside so the immune system can see what that cell is making. And that's a very efficient way to check, because cells should be making normal proteins. You've got something like 20,000 genes, but let's say your cells are only going to make, a third of those at any one time. And others will be like all the antibody-making genes. Your muscle cells, they're not going to make any of those. They're very focused on something else, and good for them. And so there will be normal proteins that would get chopped up and displayed on the surface. And your immune system learns to recognize these. Actually, any immune cells that recognize what we call self-antigen, which is basically a protein encoded by your genome that should be there, those are generally going to be killed off by the immune system very quickly. When that doesn't happen, that's when you end up with autoimmunity, which is when you make an immune response to your own, some component of your own body. And those autoimmune diseases can be terrible because it's your immune system beating you up for no good reason. And there's really not a great way to turn it off except by shutting down the entire immune system, which, yeah, spoiler alert, is bad. Is like a really bad thing to do. Yeah. So there you go. There you go indeed. So MHC molecules present. They present a little piece of generally a protein. That's just how they work. And so then T-cell receptors are only going to recognize pieces of a protein. And the things that MHC presents are generally going to be little linear peptides. So like they stretch out the protein, it like has a folded up shape, and they stretch it into a long string and then chop little bits and load some of those. And those are how you tell self from foreign proteins. Yeah. The self proteins will be things that your immune system has seen before. The foreign proteins will be things it has not seen and just happen to luckily make a response to. All right, fine. So there you go, MHC class 1 and MHC class 2. So class 1 is going to be most cells are going to make this, and class 1 is going to present to T cells that have the CD8 protein on there. And so what CD8 is it's a protein that'll reach around and it locks onto an MHC molecule. And so if CD8's locked onto the side of an MHC molecule, so imagine our antigen-presenting cell is up here. Here's our little MHC, and it's holding onto a peptide here. Let's put a peptide in there. There we go. CD8's going to lock on the side of that, and that's great. And then the T-cell receptor is going to be down here, and it's going to try to interact with the peptide. And if it can make a good match, that's great. And that T cell will get a reward and will be told to divide, basically. And if the T cell just cannot get a grip on whatever it is that the MHC is displaying, then CD8's not enough to hold on, and so there will be no reward. And the T cell will roll off and try to bump into another antigen-presenting cell. And they just do all this at random and see if it can, bind to something. Because the only way that T cells and B cells get to survive is by interacting with an antigen, being presented by an antigen-presenting cell. So the antigen-presenting cells, these are like teachers. And if it's not so much that they're teaching the immune system, they're just saying, if you can recognize this, you will get a reward. If you can't recognize it, go die, because I don't have time for you. Yeah, and the cells that recognize nothing do go off and die. Yeah, so there you go. For the most part. All right. Yes, fine. So let's look over here. So this is a class 1 MHC class 1 molecule. And so it's going to consist of a immunoglobulin-like domain down here, 2 of these little domains at the top. And you can see at the top of those domains, they're going to have a long alpha helix, which is a curvy thing, something like a phone cord. Yeah, there we go. And the peptide that is being presented is going to be sort of pinched between those 2 alpha helices. So they're gonna pinch it just like that, just like a hot dog in a bun. Yep. And then the other domain that makes this work is another immunoglobulin-like domain. It's called beta-2 microglobulin, and it's not part of the same chain. So the blue, green, and yellow chains are actually all connected to each other. They're made at the same time. And the beta-2 microglobulin, or beta-2M, comes in from the side and just anchors on here. The main chain here with the 3 domains is the one that has the transmembrane anchor. So think of this like a one-legged protein. And this is what both of the MHC molecules look like. Class 1 and Class 2 differ a little bit, and Class 2 can present larger, longer peptides. But the big difference there, which I guess I'm looking ahead, but the big difference there is that in the Class 1 molecule, it's like a hot dog bun, but with the ends sort of closed off. So there's some protein there, and so you can only fit something that fits exactly between those. Whereas with the Class 2, it's more like an open groove, so that you can fit something in that's a little bit longer, and that's okay. So there's more of a space to hold a longer peptide that can then be recognized by a T cell. Yeah. And yeah, it's again not any smarter than that for sure. Yeah. Not much smarter than that. So MHC Class 1, look at it. MHC Class 2, oh my gosh, Class 1, Class 2, Class 1, Class 2. See how those are pretty much the same? So Class 1 has this particular structure with the beta-2M, and that's our common one that most cells have. Class 2 MHC is only going to be found on certain cells of the immune system. And this is kind of a second way. So if your normal cells, if your skin cell, muscle cell, endothelium are not presenting peptides or presenting them in the right way, you have a fallback. You have a backup plan, which is for other things, like I believe macrophages, for example. So these are cells that roam around just eating whatever's in front of them, which is not a terrible life, honestly, yeah. And they will then digest a little bit of whatever they've eaten. Dendritic cells will be like this too, and they can then display it. And so these different types of cells, we'll get into which cell does exactly what down the road, partly because I have to look it up because it's hard to remember. And the only other real difference is that it's still an Ig-like domain here, but it's a separate chain. So it's 2 chains, each of which has 1 alpha helix at the top. But you see how you get to pretty much the same shape? It's just instead of doing it 3 and 1, you're doing it as 2 and 2. Yeah, domains. Yeah, so there you go. All right, so here are our 2. Here's our Class 1 on this side. Here's our Class 2 on this side. And so this is like the hot dog bun that's kind of closed. See how the alpha helices pinch a little bit toward the end? See how they sort of slope together? Because of that, it's a little tight in there, and you can only get about 8 or 9 amino acids in. And you see how this one sort of has open ends. You can actually even get a longer peptide in there if you want. This is the Class 2 presentation. And so this is all yellow because all the proteins here are coming from that main chain of the Class 1. These are yellow and green because they're coming from the 2 halves of the Class 2 molecule. And there you go. There's our little peptide presented in the groove on the top of the MHC Class 2. Interesting. Well, no, we'll save that. We'll save that for class. Yeah, but it's good. In part, we think that some aspects of human attraction are controlled by MHC. Yeah, which is weird, but whatever. Yeah. Who knows how love works? Yeah. And if you knew, would you really want to know? Yeah. You say, oh my gosh, I'm going to have to love that person forever. Well, all right. I guess it's biology, right? Okay, fine. So the concept here is anchor residues. And so what that means is that in these alpha helices, so this is our Class 1 molecule again, you can see it's kind of pinched tight. There are certain amino acids that are going to be held really close, and so they're actually going to be in position to hydrogen bond with some part of the peptide that's loaded in there. And so these are called, so the little peptides of 8 to 10 amino acids. I said the average is 9, and that's where we get that from. They're going to be held down. So there are going to be particular amino acids that can interact with the MHC Class 1, and it's the same in Class 2, molecule. And so you'll have to have just the right amino acid at this point, and this point, and this point. Basically, at both ends, you're going to have requirements. Like you're going to have a particular amino acid maybe first, and a particular amino acid maybe 8th, and maybe 9th. Other than that, the other amino acids can vary. Thing is, this limits what you can actually present to the rest of your immune system pretty sharply. Because, if a new virus rolls up, let's say, I don't know, dengue fever virus rocks up in your body, you may chop it into its pieces, and there may not be a piece that you can cut reasonably that has a proline at the first amino acid and has an arginine at the 8th position and a phenylalanine at the 9th position, or whatever it is that we need to bind to this particular MHC. And that's the thing. You get three, for class 1 MHC, so MHC class 1, you have type A, B, and C. And so you get one version of A from your mom, one from your dad, same with B, same with C. And there are a lot of versions of B out there. That's the one that's the most diverse. But genetically, you can end up like, for example, my father has, I believe, 2 of the same ones for one of his things. And it's not because he's a result of any kind of weird interbreeding. It's just that there are only so many of these different domains out there. And it's kind of luck of the draw, because I don't know how you work, but when I first started dating, people, I never did a genetic test or sequence their genome. And, your mileage may vary. Yeah. And you are biology students for the most part. So whatever. But I think there are laws around that stuff. You got to be careful with this stuff. But yeah, you could figure out, oh, okay, are my kids going to have a more diverse array of potential MHCs, or are they going to have a relatively narrow array? Because you only get 6 MHC class 1s. And if they can bind to a peptide from a thing, chances are you're going to make a good response and you're going to be okay. And if you didn't happen to inherit the right version, well, sorry, buddy. Yeah. I hope those antibodies are going to get you through, because you are not going to make a great T-cell response, and you may have trouble making an antibody response. It's going to be macrophage time, and good luck with that. So examples of anchor residues. So here we have the front of the protein, so that's our amino terminus, our engine. And here's our C terminus, our caboose, down here, so front, back. And so for a particular version of MHC class 1, you may have to have a aromatic residue here. So phenylalanine is F, Y is tyrosine, and they both have that aromatic ring in there, so the 6-carbon ring with the distributed electrons. And then they have to have a leucine, which is a fairly hydrophobic, kind of long, branchy amino acid, down here in the last position. And if you don't have exactly those 2 things, and if the peptide can't be cut to exactly the right size, then you are not going to be able to display that particular peptide. Yeah. So, it comes down to this. You can have a whole virus sitting there, and if you don't have the right proteins to be able to process it and display it, it's like that thing is invisible. And that virus or bacterium or whatever just gets to keep on beating on you until you find some way to respond. Yeah, this is why the immune system can be a little bit hit or miss. And this is the very strong individual component of immunity. And this is why you can't guarantee. You can show 2 people the same vaccine, and if one person has this and the other person has an MHC that binds this particular peptide, then they're not going to see the same thing there. They're not going to make the same response. And you only hope that you show them enough things that they can make some response. Because otherwise, things are not going to go well. They're going to say, well, I took my vaccine and I still got COVID. Yeah. Well, your genetics are partially responsible for that, and there's not a lot either of us can do about it, unfortunately. Yeah. So that's just the way it works. Yeah, it's there's a lot of luck of the draw there, and there are mechanisms, there are reasons why all these things happen, but you don't really have control over any of the parts of this. I think that's the worrying thing for me about immunology. It's stuff that you'd really like to have happen, and you just do not have a way to make it happen. Okay, fine. So All right. There's a thing called the invariant chain, and that's neat. I believe this is class 2 molecules. Yeah, there's class 2 with the open end and the little real short, looks like a 310 helix right there. So this holds a longer peptide, so you can see how long that's like our foot-long hot dog, whereas the MHC class 1 just holds the regular, inside the bun. Think inside the bun hot dogs. Yeah, sorry, Taco Bell. Let's see. MHC class 2 molecules actually start out preloaded with a little peptide. And so this little thing is called the invariant chain. And it's actually there right from the birth. This thing allows the MHC class 2 molecule to form and fold up. And at the last minute, you're going to have a way that you can actually pop that invariant peptide out and bind something else. You basically exchange it for something else. So you pop your hot dog out and put in a bratwurst or something like that, or whatever. I don't know what you like in your hot dog bun. Yeah. That's the idea. So every MHC class 2 starts out with the invariant chain in there. And there's a whole process. It starts out as a big thing and it keeps getting shorter until there's just a little bit, which is like our little thing that you can see here. That's the last step of the process. We'll get to all this stuff. All right. So MHC class 2 molecules bind longer peptides, but they still have anchor residues. And so here we can see, okay, so what do these amino acids have in common? L, F, I. So F is aromatic, but also hydrophobic. L and I are not aromatic, but they are hydrophobic. So what we're seeing here is a large hydrophobic amino acid right here will fit into the groove of this particular and interact with this particular MHC class 2 molecule. D and E are the two acidic amino acids, so it's glutamic acid and that's aspartic acid. Let's see, N, K, V, and R. The V is the odd one out there. R and K are going to be basic, so that's arginine and lysine. N, asparagine, has a nitrogen in there that's kind of positive, but it's not going to have a full positive charge. And the valine in there is small enough that I guess it is pulled back enough that it doesn't interact with whatever is interacting or trying to interact with the positive charge amino acid here. And then down here on the end, it looks like we have a little more diversity, but there are a lot of big hydrophobic amino acids in there, in addition to some that really don't fit the pattern, like D, which is acidic. It's fairly large, and so this is maybe a large space, and there may be multiple ways to interact with peptides in there. Yeah. For each of these peptides, for every specific peptide, you could go in and solve a structure and calculate exactly how the MHC class 2 binds. And we would know an additional thing, but wouldn't necessarily help you make a particular immune response because the process is still got to happen without any sort of oversight or control from your thinking brain anyway. So that's just how it is. All right, so here is a, and I'm not going to ask you to memorize this structure, but I think it's useful to show you how these things work. So at the top, we've got our alpha-beta T-cell receptor. And so you can see the variable loops, all these nice little colorful loops, purple, yellow, green, blue, light blue, and red. And you see how those are pointed so that they are interacting with, here are the helices. So there's the alpha helix on the back as behind this yellow squiggle. And the one in the front you can see right about here. The yellow squiggle is the peptide, and so it is held right in a position where it can interact with a T-cell receptor. So this is a T-cell receptor that can bind to that peptide, which is being displayed. This is the goal. This is what you want. And this sort of interaction would lead to a second signal, which would cause the T cell to start to divide and go through the rest of the steps of its maturation. Yeah, so this is a little pat on the back. If you make this thing, this is the dream right here. Yeah, but most of the time that does not happen because if you have a T cell receptor that has wonderful loops, but they do not happen to bind this particular peptide with enough force to basically hold these two cells together, then yeah, no second signal, no division, yeah, Poor lonely T cell wanders off and just disappears. Yeah, too bad. All right. So that's a MHC class 1 interaction, and you can see the beta 2 microglobulin down here. So that's our thing that's going to mark MHC class 1. It has the beta 2M at the bottom of it. And then the class 2, remember, is going to make the same shape, but it's going to do it with two proteins that each have two domains in them. So yeah, different way to get to the same point. So here is our MHC class 2 interaction. So this is still a T cell receptor, and that's fine. T cell receptors can interact with both class 1 and class 2. It's just that some cells will display antigens with class 2, and other cells can display antigens with class 1. Different types of T cells can recognize those, and so if they have the CD4 molecule, they can recognize class 2. And if they have the CD8 molecule, they can recognize class 1. And so the CD8 cells are called killer T cells. So basically they're recognizing a cell, and if they can recognize it, that means that cell is making something bad, something that it's been taught, this is a bad thing and you need to punish it. And so the CD8 T cell then kills it. It has what we call an effector function. Effector in this case being, the recipient cell dies. Yeah. And there we go. So here's class 2, different peptide, pretty much the same deal, still a T cell receptor binding to it. And so here's our alpha, which is like our light chain. Here's our beta, which is like our heavy chain. And here's our class 2 molecule with its alpha and beta chains. They use alpha and beta a lot in immunology, like a lot. It's just if you have two proteins that make up one working unit, they'll call the subunits alpha, beta, gamma, delta. They just go through the alphabet in that order every single time. So everything's got an alpha subunit, and some things have a beta, and a few have a gamma and a delta, etc. Yeah. All right. So yeah, CD4 is for our helper T cells, and there are helper T cells that can kill other cells. There are helper T cells that do a range of different things. They're probably the most complex, and the part where I think there's still a lot to learn, I believe, about helper T cells. You got the CD8 T cells, which are a little simpler, but you know what? There are wrinkles there, because they don't just go out and kill T cells. So they have been called killer T cells in the past. But they do more than that. They secrete different factors, and depending on what messages these CD4 and CD8 T cells secrete, they will call in different sets of other cells, and they'll activate them in different ways. And the T cells end up being one of the major factors that controls what kind of response you're going to make. I don't mean like good or bad response, although that is the outcome, but there are ways that the T cells and some of the antigen-presenting cells, like macrophages and dendritic cells, they're going to secrete different proteins, and these are going to bias us toward a mostly killer T cell response or toward a mostly antibody response. Or the thing that actually happens is you usually start out with one and then move toward the other. So you'll start out making T cells because, think about it, you would want to do this if you have an active infection. The T cell is going to go out there and it's going to assassinate a cell that has become infected or is malfunctioning in some way, like in cancer. It'll just assassinate that cell. That's good. That's efficient. Antibodies are like for cleanup. So any other, virus that happens to be floating around somewhere, you can make an antibody, slow it down, allow it to get destroyed, and that makes things better. But antibodies are not usually what you want as your sort of first response to an attack. Antibodies will, though, be secreted on the mucosal surface, that IgA, and so antibodies can prevent the attack by a foreign invader from even starting. So I guess antibodies are your first and your last line of defense, and in the middle, you want to be pretty T-cell focused in order to make a good response. And if your body makes whatever the appropriate response is for the thing that you're facing, you probably don't get sick. You may not even know that you're infected. We run into pathogens every day and quite often do not even realize it because our immune system just deals with it. And if you don't feel sick, you don't know what you happen to have breathed in because there's so many viruses. Let's see, a cubic meter of air has somewhere around 1 million to 10 million individual virus particles in it, all the time, everywhere, all day long. Yeah, they're mostly not human viruses, but a couple of them are, particularly if you're in a room full of people, like a classroom. And so, yeah, one person breathes it out, you breathe it in, whatever. That's how bad things happen. Yeah. Okay, so this is what the CD4 and CD8 molecules look like. They both have immunoglobulin-like domains. They're both pretty tall, so they're actually going to stick out. Your cells are going to have this thing called an extracellular matrix, which is like I don't know, it's a gloopy, sugary, proteiny sort of coating on the outside. It's like a little sweater that your cells are all wearing. And these things are just tall enough that they can poke out a little bit from inside the little cotton balls that are covering the rest of your cell. It's not actually cotton, obviously. It's extracellular matrix. And so CD4 has 4 immunoglobulin-like domains, and CD8 has 2 immunoglobulin-like domains. CD4 is just 1 chain, CD8 is 2 chains, and whatever, yeah. There are 2 different ways of doing a thing. So CD8 is going to have the right stuff to interact with an MHC class 1 molecule that has already come very close, that's already been kind of snared. And CD4 will be able to interact with an MHC class 2 molecule. Yeah, there you go. That's the deal. And these are both going to be expressed on T cells, but generally T cells are going to have one or the other. Or rather, that is the most common situation. There will be cells in your gut, though, and various other places in the body that will actually make both CD4 and CD8 T cells and have important roles in, allergy, for example, or tolerance. The reason why you can eat a sandwich and not make an immune reaction against the sandwich, usually. But of course, when you do make that reaction, those would be food allergies, and yeah, there are mechanisms behind all of this. So here you go. There's CD4 interacting with MHC class 2, and we haven't drawn a little peptide in there, but you got to imagine it in that little slot at the top. Here's our CD8 interacting with MHC class 1, and the only thing we're missing here is that if this is a T cell, it's going to have a T cell receptor. So that's the other protein, and it's going to come out here and sit boop right in that space, so it can also interact with kind of the other side of the same peptide. So class 2 is going to be holding on to, let's think of it as the bottom of the peptide, and the T cell receptor is going to try to bind to the top surface of the same peptide. So they can't bind exactly the same thing. They've got to bind opposite sides of the same thing in order to make this little immune sandwich that we're building right here. You'd say, oh, that doesn't seem terribly probable that this would happen. And you're right. Yeah, this is unlikely, but it happens just enough that it keeps us alive. And the immune system is very good at finding needles in haystacks, so to speak. So once the immune system has found something like this that works, then it will make lots and lots and lots of copies of that particular T cell very quickly, so that each one of those can go out and try to help you, because it is rare when you get a hit that's successful like this, and it takes time. It takes a lot of trial and error, because that's the guiding mechanism behind all of this. So here's the entire sandwich. This is the whole enchilada. So we got our alpha beta T cell receptor. So this is our standard T cell receptor on a normal T cell. What kind of T cell is this? Well, if it's got a CD4 protein on it, we can think of it as a helper T cell or a CD4 positive T cell. Yeah. And there are several subtypes of that because, of course, there are T cells. They're the whole rainbow. Here's our MHC class 2 molecule, because that's the one that CD4 and cells can interact with. And I don't see a little peptide there in the groove, but I'm assuming it is there somewhere, or else that's where it would be, at least in there. Yeah, fine. Here's the whole deal for class 1, because just to show you how this works. So there's our T cell receptor, bam, and so our T cell is going to be on the top, and the T cell is going to have a long sort of string of protein reaching down here. So this is where CD8 is going to bind, actually toward the bottom of the MHC class 1. But so that's why CD8 has to be on kind of a long stem. And yeah, the two of these are going to work in tandem. If the CD8 can lock the MHC class 1 in position, then the T cell receptor can try to bind and see if it binds. But if the T cell receptor can't bind, then CD8 is not going to be able to hold on for long. Here's our little peptide. It's this little squiggle in the center, because there's the alpha helix on the one side, and here's the alpha helix on the other side. And this yellow domain is, of course, the beta-2 microglobulin, a little tiny immunoglobulin-like domain that fills in the little missing second leg. And remember, beta-2M does not connect to the membrane. It's just floating there. It's the main chain, the alpha chain, that actually does go through the membrane. All right, so let us look at, so this table I would say is worth learning because you will need this down the road. This is a good thing to look at and think about, and you need a mental picture of what this is saying. So MHC class 1. So I told you it's on pretty much every cell. Answer is it's pretty much on every cell. So you'll get some on T cells, and you'll get some on various other tissues. Like all cells basically have to make a little bit of MHC class 1 in order to be cells, except for red blood cells, which don't even have a nucleus. And probably it's kind of questionable about whether we should call them cells per se, because they don't get to divide, and yeah, they're remnants of a much larger cell, which the stem cell that made them. They're kind of like a big platelet, honestly. But yeah, whatever. Their name is red blood cells or erythrocytes, so red cells they are. Yeah. But you can see there's a ton of MHC class 1 on some of these cells. So T cells and B cells and macrophages are great at presenting a little bit of whatever they're making. And so this is like the police policing themselves, maybe. So we have a really strong sort of surveillance on the actual immune system, showing a little bit of what it's making. And cells from the immune system are going to move to areas like lymph nodes, for example. And so they're going to be in contact with lots of other cells of the immune system. And so that's a good thing. If they happen to be able to display a peptide, they can be both antigen presenting and antigen recognizing cells at the same time, which is really cool. Yeah. Okay. T cells can recognize MHC class 2, and the little star says that in humans, they can rather express MHC class 2. In mice, all T cells do not express MHC class 2. So, but whatever. We're not here to talk about mice. So just learn the in humans thing. B cells express a ton of it, and dendritic cells are great at presenting both class 1 and class 2. So dendritic cells and macrophages are what we call professional antigen presenting cells. This is their main function, and it's most of their function. They present to T cells and B cells, and they interact with those T cells and B cells to tell them if they have made a good match or have not made a good match. Red blood cells don't do anything, then they kind of aren't anything. And other cells in the body will not express MHC class 2, except for microglia in the brain, which are kind of like macrophages. So things like astrocytes, I believe, would be included with this, which are kind of like a macrophage-like cell. So the brain kind of has its own version of the immune system, which we don't get into as much. And it's sort of walled off from the rest of the body. You have this thing called the blood-brain barrier, which basically keeps your normal immune system out of the central nervous system. And so you have like a separate system in there, but mostly it relies on making sure that things do not get into the central nervous system. So That's the job of the main somatic, if you like the whole body immune system. Yeah. And then you've got extra little cleanup crew in the brain that can sometimes help and sometimes make things worse. And if you think about it, I think that makes sense as well, because whereas you can blow up a muscle cell and the muscle will still work, and you can destroy, a liver cell and repair it from a stem cell, if you lose a particular neuron that was connected to other particular neurons, you may not be able to build all those connections again. And so the sort of immune response that we make, where there's a big focus on killing infected cells, can be very devastating when it's in the brain. And so you'll see when you get virus and bacterial infections in the brain, those can have pretty severe consequences, because if the only way to kill the pathogen is to kill your brain cells, then you're going to lose, potentially sight or hearing or, any of the other things. You may even die. So very dangerous when something does get into the brain. And yeah, definitely that is not the thing that you want to happen. Okay. So there are a bunch of MHC class 1 like molecules. So one of these things is going to be called CD1, and it looks like MHC class 1, but it tends to present lipids and carbohydrates. Yeah, and there are other versions of this thing. That's fine. In addition to those normal cases, we would also have these other types of T cells. So these are called gamma delta T cells. And so instead of making the normal T cell receptor, which has the alpha and beta chains, they make a T cell receptor which has a gamma and a delta chain. They look just about the same. They have variable regions on them, kind of like normal. But the gamma deltas tend to interact with these alternative presentation proteins, so these non-classical MHC class 1 like molecules. So they're not really MHC class 1, they're just built kind of the same, and they have a beta 2M, but it's a different chain. It just happens to fold up in the same way and do a very similar job. So this is one of those molecules called T22, which is shown interacting with a T cell with the gamma delta T cell receptor. So the T cell's on the top, the antigen presenting cell is on the bottom. Yeah. All right. And yeah, stuff in blue won't be covered, but that's just in the in the sort of lists at the start of each section. So there you go. Yeah, I hope that helps, and we'll cover more of this in class. And between these things and the book, yeah, the book is good. Just read the book as well. Ask me whatever you want. I'll do my best. I don't always know the answers. Sometimes I can look it up. I will do my best. All right, thanks very much. This has been Chapter 4, Immunology, BIOL 454. Yeah.